for kind words, and thank you for the introduction, Dr. Gattani. It's pleasure. Um, Today I'm going to talk about stem cell modeling of endometriosis and its application in disease modeling. Let me share my screen here. Okay. So, is it visible?
Yes, sir, it is visible, but
Okay.
not in the full screen mode.
Oh, sure. Yeah, nice.
Yeah, okay, sir.
Okay. So, okay, so I am currently working at a Henry Ford Hospital in Detroit, in Michigan. Earlier, before I joined here, I was a postdoc at Wayne State University, as Dr. Gattani mentioned. And in my first postdoc at Boston Children's Hospital, I worked on um, modeling endometriosis disease, which is a gynecologic disorder and characterized by chronic pelvic pain and infertility. So today's talk will include a basic introduction of embryonic stem cells and what are induced pluripotent stem cells. And then I will talk about differentiation, which are two types, spontaneous and directed differentiation. Then I will cover briefly applications of IPSC modeling in disease. Um, and then from my first postdoctoral work, I will detail endometriosis modeling, which I did using murine embryonic stem cells. After that, we'll go to conclusions and finally acknowledgements. Okay. So, pluripotency is cell's ability to differentiate into all the three germ layers, which are ectoderm, endoderm, and mesoderm, and all the organs derived from these three layers that result in generation of the whole organism. And these pluripotent stem cells can be classified into two types: embryonic stem cells or induced pluripotent stem cells. Embryonic stem cells um, are the cells which are derived, which are present in early embryo in the inner cell mass of blastocyst, which is a stage after fertilization, and then zygote, then eight-celled stage morula, and then blastocyst stage is generated. So embryonic stem cells are the stem cells which reside in the inner cell mass of this embryo or blastocyst, which are retrieved and cultured in vitro to generate to produce pluripotent stem cells. On the other hand, the induced pluripotent stem cells are not embryonic, are not of embryonic origin, but instead they are the samples, tissue samples, maybe fibroblast from skin, blood cells, or from urine, some cells. They can be programmed and they can be induced or administered the transcription factors that can then lead to generation of this pluripotency in those cells, and thus they result in induced pluripotent stem cells, and they can also give rise to all the three germ layers as well as the organs derived from those layers. This reprogramming or induced pluripotency generation was discovered by uh, um, two biologists who got Nobel Prize also for their work, Takahashi and Yamanaka. In 2006, they demonstrated that if we transduce OCT4, SOX2, KLF4, and c-MYC, these are transcription factors. If they are transduced into the cells of skin or blood origin, um, and not embryonic origin, if they are transduced to reprogram this, then it would be sufficient to reprogram these fibroblasts from mouse or human or any other organism to embryonic stem cell like identity and so-called induced pluripotent stem cells. In short, I will call it IPSC. So this picture here shows these four or five different transcription factors that can be genetically induced or genetically introduced in these fibroblasts. Um, and therefore, they are reprogrammed and they then become pluripotent stem cells from which we can get these cardiomyocytes, the cardiac cells, adipocytes, fatty cells, and neurons, neural cells like these are the axons and motor neurons, also astrocytes, glia cells, which are of the brain origin. These are pancreatic beta cells, the islets of Langerhans in pancreas, as well as hematopoietic progenitor cells. So this discovery was a revolution, and it revolutionized the research world. How that if for a disease, if we want to study a disease mechanism, and we it is hard to sometimes harvest or to collect cells, the diseased cells, due to its low number. And from those patients, if we can get the adult fibroblast cells and we can reprogram those cells and then generate the diseased cell phenotype after reprogramming, those diseased phenotype cells can be then studied and um, interrogated for the aberrant molecular mechanisms associated with that particular disease. Now, what we can do with these induced pluripotent stem cells, that either we can spontaneously differentiate them, so without any inducers, without any growth factors added, if we leave them without leukocyte inhibitory factor, so LIF, which prevents the differentiation of these maintained cells, pluripotent stem cells. So in the spontaneous conditions without any growth factors, if they differentiate into these three germ layers, uh, uh, we will get non-specific lineages of germ layers and cells, because we don't know, we will have all the three mixed germ layer derived cells in those. On the other side, there is a directed differentiation protocol, and in this in vitro technique, we add small molecules or morphogens or growth factors. We also call them inducers. And what they do after we add them, they are involved in patterning. They direct, or they specify and commit these cell types to undergo a definitive lineage, and they can generate just the organ or just the cell types that is determined by those growth factors added, because those growth factors act on a specific signaling mechanisms, and due to which they differentiate into the specific lineage of cells. Okay, so like I was telling earlier, that if we get these somatic cells or fibroblasts cells from these patients, we can then reprogram and in vitro differentiate these iPSCs into the disease relevant cells. Um, like suppose, for example, um, in diabetes, we get these cells from patients and in vitro differentiate Hello, hello,
Hello, sir.
hello.
I think,
Sorry about that.
hello, I
I
think there
don't.
was some problem.
Yeah, that's
The
why.
the internet connectivity. So sorry for disturbance. So yes, sir, you can go. Let me make you the co-host.
हाँ मैडम कल है क्या अभी अभी है
Yes, sir.
Okay, is it working now?
Yes, sir.
Okay, so I will. I mean, this slide I think I covered. So okay, yes. Okay. So all right. So are you able to see the slides?
Yes, sir. It is visible.
Okay, okay. So yeah, I was just talking about this disease research applications that after we get these disease relevant cells and from these IPSCs, we can check the targets and we can validate the targets, and accordingly we can design a drug also for that disease. And it will be compared like in parallel, side by side. So hello, sorry, was there any question?
No, sir. I think it is by mistake.
Yeah, but if there is any question, I'm happy to answer. They can interrupt me anytime. It's totally fine. Okay.
Okay, sir.
Yeah. Okay. So and then we can also, based on these target identification, the gene editing can also be done in these cells, and we can genetically correct those cells and look for the reversal of the disease symptoms or disease related mechanisms if they are corrected in those healthy cells after gene editing. So seeing this, it is a powerful tool that iPSC derived cells from diseased from patients, and then how it can be used for drug development as well as the cell therapy applications. That's a tremendous benefit of the iPSC and stem cells. Now I will switch to my work that I did during my first postdoc at Boston Children's Hospital or Boston Center for Endometriosis. Um, so, like I was introducing in the beginning, that endometriosis is a gynecologic disorder of women that results in chronic pelvic pain and infertility. Before we move to the endometriosis disease itself, let me just briefly go over this anatomy of endometrium or uterus here. So inner layer of uterus, which is known as inner membrane of uterus is divided into three layers. Sorry, I have to. Yeah. So, like the outside layer of the uterus is perimetrium, then middle layer myometrium, and inside is the endometrium. This inside layer, endometrium, is very highly vascular, simple columnar epithelium, and it is ciliated and contains a lot of secretory cells. Um, Also, it has uterine or endometrial glands. So during menstruation, each month, this layer regenerates as due to shedding of the regenerated layer, the outer layer, superficial layer. Sorry. So I'll go back. I'll step back. And so this endometrium again is divided into two layers. stratum functionalis that is shed during menstruation, each menstrual cycle. And stratum basalis, that is basal layer, which gives rise to functionalis. So after shedding of this functionalis, the stem cells which reside in the basalis layer, they give rise to this functionalis layer again. And that's how the um, uh, um, that's how the menstruation happens. So what happens in this endometriosis disease, um, that these, so this is a uterine structure here, and these spots here, they are these endometrium, which grows outside its normal location. So normally it is inside this uterus, but in endometriosis, it grows outside, maybe in the ovarian region or in the peritoneal region, and it leads to pelvic pain and infertility because it undergoes bleeding and shedding every month, just like the normal inner lining of the uterus, and that causes the pelvic pain with each menstruation. So that is the reason that women cannot actually uh, dis distinguish whether it's endometriosis which is causing the pain, or which is or whether it is the regular uh, 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 puberty or or the periods that is causing pain. So that's why a diagnostic delay is there. Now, this occurrence of ectopic endometrial, so like growing of this endometrial layer outside its normal location, which we call ectopic endometrial glands and stroma, they respond to cyclic changes in estrogen levels and result in inflammatory processes. About 5 to 10% of women are associated with endometriosis, and they experience the painful intercourse, painful menstruation, painful urination. Now, okay, the molecular mechanisms which are involved in endometriosis, there have been several theories which have been proposed, like the one which are which are important. I have boxed them in these rectangular boxes, like short menstrual cycle length, or uh, estrogen receptor beta expression is regulated in the endometriosis patients. Hoxside 10, these are genes like nuclear factor kappa B, matrix metalloproteinases, MMPs, protein kinase B. So several of these genes have been studied, and they have been implicated in inflammatory response, as well as increased sensation of the pain receptors. But none of these have been proven to be definitive mechanisms, and there is no definitive drug or therapy yet available for this disease. The only surgical treatment is laparoscopy and surgically removing these outside endometrial lesions. Okay, so in this project, we used mouse embryonic stem cells of G4. So G4 is the name of that mouse embryonic stem cell line that that we used to model endometriosis. The rationale behind it was, like, ESCs are self-renewable cells with the capacity to differentiate into any tissue types, making them an invaluable tool to study mechanisms of pathogenesis through disease models. And the ability to produce endometrium from these stem cells in vitro, it offers a way to investigate both normal and ectopic endometrial tissue development and identify endometrial progenitor cells. So how we used it, this slide just shows a methodology that we cultured these mouse embryonic stem cells in 10 cm petri dishes, and then and then we removed this leukemia inhibitory factor, LIF. What happens if we remove this factor? The pluripotent stem cells are no longer undifferentiated, and they start differentiation. So we left them in suspension for about 2 weeks, and after that, they make clumps among themselves, which we call embryoid bodies, and they look like three-dimensional aggregates of these stem cells. And then these embryoid bodies we recovered and attached them on gelatinized plates. And these, during this, after attachment, we induced the differentiation of these EBs into endometrium-like cells, which comprised of both glands and stromal cells. And the growth factors that we used was activin A and bone morphogenetic factor, or bone morphogenetic protein BMP4. And after that, when we after that differentiation, we wanted to look at the expression of these different markers of endometrium. So we used immunofluorescence, fluorescence activated cell sorter or flow cytometry, and quantitative real-time PCR to look at the markers. So these are some characteristic findings. So because these were mouse embryonic stem cells, we first looked at the mouse uterus itself, because we wanted to see whether this mouse uterus, uterine lining or membrane, has the expression of all these markers which are found in human endometrium also, because we are going to model human disease in mouse embryonic stem cells. So we have this HND staining here just for the nuclear, then this cytokeratin, which is which stains the cytokeratin or cytoskeletal marker in the cells. This is vimentin marker, which is for the stromal cells. Then E-cadherin in the green lining, it stains the glandular epithelial cells, cell marker. And then estrogen receptor A and B, which shows up nicely. And then progesterone receptor A and B. Then CD9 and 13. So CD9 is the glandular epithelial cell marker, and 13 is the stromal cell marker. And then these CD146 and PDGFR beta, which are the mesenchymal stromal cells or mesenchymal stem cells, which are found in the human endometrium. So we looked at, and they are found around the blood capillaries in these endometrial-like cells. So we were able to see those. And then HOX-10, so homeobox protein, which is another marker for development of uterine embryonic development. And HOX-10 was also stained positive. This is a sketch here that how this endometrium layer looks like I was explaining in the previous figure. So this is a myometrium, which is the middle layer of the uterus, and then inside layer again is divided into basalis, where the bases of the glands, endometrial glands are there, and the perivascular cells, this CD146 and PDGFR beta co-expressing cells around the vessel lumens. And in the functionalis layer, these stromal cells are found mostly, and the apical parts or top parts of these glands are found. So every menstruation cycle, this functionalis layer is shedded, and the basalis layer helps regenerating it back, regenerating it every cycle. So after that mouse uterus characterization, we then differentiated our mouse embryonic stem cells using those two growth factors, activin A and um, BMP4. But here you can see, so first column is the DAPI, which stands nuclei, and then second column is CD9, third column is CD13, and the last one is the merged image of both. We differentiated these cells for 3 weeks, and every week we check the expression by immunofluorescence. And we can see, like this bottom panel shows here, that initially when we differentiate just only the embryonic stem cells, we saw that these CD9 and 13 are co-expressed like all the cells. So a cell which is expressing CD9 is also expressing CD13. So we can say that in the early periods of development, um, both the markers are co-expressed, and later in the course of third week or fourth week of differentiation, which I will show in the next slide, these markers then separate. So in the we can say that this model, this in vitro system, is like early reproductive, early endometrial generation model also. And then, like I mentioned, when we looked for the expression, so this was just the embryonic stem cells without making the embryoid bodies, the three dimensional aggregates. This was the expression pattern. But when we generated embryoid bodies out of those embryonic stem cells, and um, um, again directed their differentiation further after 3 weeks, we were able to see that CD9 and 13 separated really well. Although the percentage of co-expressing cells went down, and the HOXI10 was also expressed by CD9 positive cells as well as CD13 positive cells. So HOXI10, because why we chose HOXI10 here, HOXI10, because these CD9 and CD13, although they are markers of uh, endometrial glands and stroma, but they are also expressed by other cell types in the body, like kidney tubules. They also express it. Therefore, in addition to CD9 and 13, we also included other markers of embryonic development or uterine endometrial generation, which is homeobox 10 or HOXA10 marker, as well as these mesenchymal stem cell markers. The bottom picture shows here the gel. So this was the RT-PCR that we did after collecting RNA from these cells. So these cells were sorted using FACS, and then just the and then looked at the markers of glands, stroma, and other markers of endometrium generation, like HAND1, FOXA2, estrogen receptor A and B. The last one is negative control, and the beta-actin is positive control. And we were able to see that all the markers were expressed in these differentiated cells. So these, what later we thought that we will sort these glands and stromal cells, so CD13 positive cells, and also the dually um, co-expressing the cells which co-express CD146 and PDGFR beta. We sorted them using FACS, and then looked for the different markers of endometrium by qPCR, and we were able to see that CD13 positive, CD13 positive cells, the stromal cells, differentially express the estrogen receptor A and B, progesterone receptor, HOXA10, FOXA2, and HAND1. So these all genes are specific for our endometrium generation or endometrial cell type markers, which suggests that in our in vitro mouse embryonic differentiated cells, we should be able to, or we developed an in vitro system to study the human endometrium generation, so that we can manipulate genetically and stop the generation of endometrium in future, and which could be an alternative strategy or therapeutic strategy for endometriosis disease. Now, we also looked in these sorted cells by microarray, and we did the ingenuity pathway analysis and IPA network. It showed all the markers, including ERA, ERB, PRA, ERB, NF-kappa B, matrix metalloproteinase 3, VEGF, which is a marker for vascular integrity, and Hoxaten G in these differentiation. So, in conclusion, I would like to state that these murine embryonic stem cells have the potential to differentiate into endometrial precursor and mesenchymal stem cells. And it is an in vitro system to model early endometrial tissue development, a key step in elucidating the mechanisms of ectopic endometrial tissue growth. Also, we can develop strategies to prevent endometriosis, we can establish novel therapies. So the future goal was to use CRISPR technique to knock down one of these markers of endometrium and see if we can stop the regeneration of endometrium from mouse embryonic stem cells, as well as identify approaches for non-invasive monitoring of disease progression. And this was the reason we used mouse embryonic stem cells, because from patients, it is very hard to get sufficient amount of endometrial stem cells. However, the fibroblasts can always be used to be reprogrammed and then later on to generate these endometrium tissues. But we did not pursue that approach. So this is why this whole project shows the importance of stem cells, how important and how critical these stem cells can be, and we can study the key signaling pathways in these IPSCs derived from these diseased patients. With that, I would like to acknowledge my supervisor at Brigham and Women's Hospital, Dr. Raymond Anchin, and this was the whole team then. It is like it was in 2016 when I pursued this work, and these are research assistants, these are research associates, and so Dr. Stacy Mismer, Dr. Mark Laufer, Nicholas, Emma, Sophia, and all other teams, and of course my employer. So along with that, after concluding that, I would like to take any questions that you may have. Thank you.
Thank you, sir, for your nice presentation. So I request the participants who can ask the questions with Dr. Parashar.
Yeah, feel free to ask anything that you think was confusing or was.
Actually, one participant asked about, please share the PPT. So I would like to inform you that all the session, they are being recorded, and these recorded session will be available on YouTube. So you can see later on also. And the SAM link will also be provided to the ATAL FDP. So when we finish the FDP, then we will edit the things, edit the videos lecture wise, and we will upload over the YouTube, even on our university website also. So any more question?
Hello, sir. Sir, excellent presentation. I just had one question. In your
Thank you.
first slide, you showed how the fibroblast cells were converted into the stem cells. Sir, during the
Yeah.
creation of Dolly also, I think they did something quite similar. They had converted the mammary cells into totipotent cells and recreated. So that was before this, I think. So how is it different? They had fused it with the ovum cytoplasm.
Right. Yeah. In case of you are right that in case of Dolly, it was the somatic cell nuclear transfer, and
Yeah.
that was the nucleus from the
Mammary
from the
cells.
memory cell.
Hmm.
Yeah, it was injected into the ovum. Yeah, and then later on it was induced to generate into the blastocyst stage,
Yes.
right? And then developed into the clone itself.
Hmm.
So I think at the 8 or 16 celled stage, that embryo itself was re-implanted back into the surrogate.
Yes. Yes.
Yeah, and then after the full gestation, the cloned Dolly was given birth. Yeah. So that is different in a way that I think they did not, but
Yes, sir. I guess they did not know the transcription factors. They had just somehow
no,
used
they did
the
not reprogram
cyto.
anything here.
Yes.
Yeah,
Yes.
but
Here
it was
they did.
just the, yeah, and
Ah,
in this Yamanaka, like,
yes.
where
Yes.
is that
Yes.
slide? Sorry, it's in the back. So in this one,
Yes, sir. This
they
slide.
used the